Hey guys, so I just wanted to make a quick video to go over the Unit 3 FRQ. I thought this one was kind of difficult, so I wanted to make a video for you guys to watch if you were struggling with it. And feel free to still text me or set up a virtual conference to go over it together or to check your answer to make sure that it works. But again, I was just going to kind of talk through it and let you guys know kind of what I thought as I went through it and how to best answer these questions. So first of all, let's just do a brief overview of cellular respiration. If you go to modules and we look at our Unit 3 module, I have a lot of videos up here that you guys can use to review. We have the College Board review videos of the enzymes, photosynthesis, cellular respiration. Also, these live stream videos are put together by AP Bio teachers that go over all three of these in one video. And then right here, I have this thing that says great resource for reviewing cellular respiration. So if you click on that, it's going to bring you to this document that I just went ahead and opened in another page. So I just clicked right here and it brought me to this. So I really like this document. I can't take credit for putting it together, but I found it and it was really helpful. It tells you all the reactants, products, and diagrams, and then the special notes about each step. Like glycolysis, my favorite part is probably the notes that are added. No oxygen needed, occurs in the cytoplasm. Also, again, when we get into evolution, we would talk about how early Earth had a low oxygen concentration, so we could reference that glycolysis could have developed before Earth had oxygen. Again, that's an evolutionary perspective, but again, it's important to know in the context of AP Bio. I don't know if they would go there since we didn't get into evolution, but it's a good note to read. Again, these little notes are all really helpful to kind of go over what happens in each step. I like the notes on the electron transport chain that tell you that you remember that this is a stage where we need oxygen. It's called oxidative phosphorylation, and this is where the majority of our ATP comes from. So if you guys remember cellular respiration, the whole point is to break down glucose. And so if you remember, the thing I had you guys write down is every time you break down a molecule, electrons are released, picked up by electron carriers, and brought to the electron transport chain. So that's just the thing that is the most important sentence to remember when you are thinking about cellular respiration. At every step, we break down a molecule. Every time we break a bond, we release electrons. Those electrons are then carried via electron carriers to the electron transport chain, where we can use all those electrons to make ATP. And again, if you want to look back over what happens in the electron transport chain, as electrons pass through the proteins in the membrane, we pump protons out, creating a gradient. We call that a proton motive force. And then we let those protons back in along their gradient to create ATP. So the electrons are carried via electron carriers. They travel through the membrane. As they release energy, we use that to pump protons out. And then when we let the protons back in, we create ATP. This is another step I really liked here. It said some vocabulary, substrate level phosphorylation versus oxidative phosphorylation. Those are always confusing for students. Also, the proton motive force, which I just referenced, is the force that promotes movement of protons across the membrane to generate a gradient. And we can sometimes call the production of ATP from this gradient chemiosmosis. So if you ever see any of those terminology, don't be intimidated. It's all just what's going on right here in the electron transport chain. So here's our summary diagram. It starts with glycolysis, where we start breaking down glucose into our pyruvate molecule. As we break that bond, we're going to release electrons that are carried via electron carriers to the electron transport chain. Pyruvate is further broken down into acetyl-CoA, which is necessary to enter the Krebs cycle. Then as we move into the Krebs cycle, we break down even more of the remaining glucose that was the carbon that was originally found in glucose. And as we break it down, we release more electrons, which are carried to the electron transport chain, which we use to make ATP. So let's get to the actual FRQ. This is a long one. This is question 1. So remember, you guys will have a question 1 on the actual exam. There's going to be a question 1 that is going to be experimental design based, and it will tie in some type of concept from AP Bio. And then there's one that's more conceptually based. So this is the long one, and it's worth a little bit more on your actual exam score than the other one too. Electrons from oxidation of glucose. Oxidation is the breaking down of glucose and other nutrients by glycolysis in the Krebs cycle are added to the electron-carrying compound NAD+ to form NADH. So when the electron carrier picks up the electrons, it turns into NADH. So a lot of times I reference the electron carriers as like Ubers. So this is the empty Uber, and then this is it when it has an electron in it, shuttling it to the electron transport chain. Once it drops off its electron, it would convert back into NAD+. So once you drop off your passenger, you're an empty Uber again. To meet the continuous requirement of NAD+ by cells, NADH is reduced back to NAD+ through the electron transport chain in the presence of oxygen through fermentation in the absence of oxygen. MTND5 is a mitochondrial gene that encodes a subunit of NADH dehydrogenase. Remember, anytime we end in A's, it's probably an enzyme. This enzyme catalyzes the initial oxidation of its substrate, NADH, to NAD plus NH in the electron transport chain of mitochondria. So basically, this enzyme is going to be responsible for taking the electron and returning this molecule back to the empty Uber and a proton, which we can pump through the membrane to create ATP. A researcher hypothesizes that the mutated NADH dehydrogenase, so the enzyme is mutated, has decreased activity but is not completely non-functioning, and that by increasing the pool of NADH in cells, the activity of NADH dehydrogenase will increase. So we're talking about enzymes here. So remember, in enzymes, we have a substrate and we have an enzyme. So here, our enzyme is NADH dehydrogenase and our substrate is NADH. So the NADH dehydrogenase is the enzyme that is breaking down the substrate NADH into NAD plus NH. So if we have, let's see, the NADH dehydrogenase, it's a really long word. That would be your enzyme. The substrate that would fit into it, we'll make that a different color, is NADH. And then our products, what's going to be produced after that breaks down, it's going to turn into NAD plus and H plus. So here our green is our enzyme, our substrate is the red, and then our products are the yellow. So just kind of keep that in mind as we read through it. To test this idea, the researcher treated a group of individuals with the disorder with a vitamin that is similar to NADH and measured the concentration of NAD plus and lactic acid in the blood over the course of 20 weeks. The results from the representative individual are shown in figure 2. So basically we're saying we're finding something that's basically the same shape. It acts like NADH, and it's going to create NAD plus and H plus as well. So we're just pretending that they're testing NADH, but instead it's actually a vitamin. It's just the same shape, so it can fit in the enzyme and produce the same stuff. All right, why do you not want to scroll? Okay, I know why. All right, the concentration of NAD plus and lactic acid in the blood are representative of the representative treated individual. So here we have the concentration of NAD plus is being measured. It increases, and then it's eventually going to decrease over time. And then our lactic acid concentration is going to decrease over time. So as we have more NAD plus, we're going to have less lactic acid. So as we are able to break down the NADH and return it to the electron carrier, it can go pick up more electrons and continue the process of cellular respiration. We have less buildup of lactic acid. Describe the pattern of inheritance that is most likely associated with a mutation in this gene. Explain why individuals are not typically heterozygous with respect to mitochondrial genes. So it tells us it's a mitochondrial gene. This isn't something I was able to go over a lot because it was in the little online unit that you guys watch videos on non-Mendelian genetics patterns. But it's important to know and make yourself a note of that mitochondrial genes and mitochondria are inherited through the mother. So they're maternally inherited. So describe the pattern of inheritance most likely associated with that. If it's found in the mitochondria, it's probably mitochondrial inheritance that is inherited through the mother. And explain why individuals are not typically heterozygous, because you only get one copy of that from your mother. And so it's not a paired chromosome, it's just an individual chromosome. The reason is the egg is a lot bigger and it has a lot more organelles in it, and the sperm is pretty much just a packet of DNA, haploid DNA that's being driven by a little flagella, and once it fuses with the egg, you retain most of the organelles that were in the maternal egg cell instead of actually using the organelles that were in the sperm. And so people get their mitochondria from their mom, so any mitochondrial disease would come through a maternal line. And again, you only get one copy of those because you don't get a copy from your father, so that's why you only have, you're not heterozygous, you only have one copy, you don't have two. Identify a dependent variable measured in the researcher's experiment. So remember, our dependent variable is what we're measuring. So what are we measuring here? The scoring guidelines accept two different things that you could say for the dependent variable. You could say the concentration of NAD plus or the concentration of lactic acid. Both of them are being measured, so either answer would be correct. Okay, so we identified the dependent variable. Again, either one is accepted. Identify one control that a researcher could use to improve the validity of the experiment. So they're trying to see, how do people that have a mutated allele, MTND5 gene, so how do people with a mutated MTND5 gene break down NADH and turn it into NAD plus, and how does that affect their lactic acid concentration? So we're studying individuals that have a mutated gene and seeing how they break down NADH. So what could be a control for this experiment? Well, instead of using people that have a mutated gene, we could use a reference of people who do not have a mutated gene for comparison. So I think the best control there would be to use a person that does not have a mutated gene and see how they break down NADH and compare the graphs to see if they're doing it differently. Justify the researcher analyzing blood samples at many intermediate time points instead of only at the beginning and the end of the 20-week period. Just more sampling is going to give you more reliable data. So that one's not too deep, just knowing that taking more and more samples are going to give you more and more reliable data and a more reliable graph. All right. C, describe the relationship between the concentration of NAD plus in the blood and the concentration of lactic acid in the blood during the first 5 weeks. So if we look here from 0 to 5 on the graph, that's where our increasing NAD plus is happening. And from 0 to 5 on the graph here, we have decreasing lactic acid. So as NAD plus is increasing, lactic acid concentration is decreasing. Based on figure 2, calculate the average rate of change in blood NAD plus concentration from week 5 to 17. So we're looking at this graph from week 5 to 17. They want you to find the rate, and remember the average rate of change is just your slope. So you would do y2. That's so great. Mommy's almost done, baby. Y2 minus y1 over x2 minus x1. So you would find the y value at 5, which is about 300, and you would subtract the y value at 17, which looks like it's about 75, and then your x values would be 17 minus 5. So let me show you guys. Sorry, guys. Hold on. Sorry, I was a little distracted there. I think I might have said it wrong anyway. So we have x2 and x1. So our x2, baby, you have to stop talking. No. All right, guys, sorry about that. So if we look at the slope formula, it is y2 minus y1 over x2 minus x1. Our y2 is going to be the value of whatever the 17 minutes is. So that looks like it, again, is about 75. Our y1 is wherever the y value is at 5. So that's approximately 300. Our X2 was the value at 17. And then our X1 is our 5. So you would just plug the values in and you would determine the rate. And remember that you don't get credit for a rate unless you include the units. So our units would be the concentration of NAD plus over time. Okay. The researcher performed a follow-up experiment to measure the rate of oxygen consumption by muscle and brain cells, predict the effect of MTND5 mutation on the rate of oxygen consumption in the muscle and brain cells. So if the mutation is preventing the NADH from being properly broken down into the electron carrier and the proton, then it's going to slow your rate of cellular respiration because it's going to affect our electron transport chain. So if we look back here at our electron transport chain, this is the protein we're talking about that's able to convert NADH into NAD+. It's going to take our Uber that has an electron, it's going to take the electron away, and it's going to regenerate the empty Uber that can go get more electrons. So if we are preventing this step from occurring, where we're unable to regenerate the empty electron carrier to go get more electrons, then the whole electron transport chain is going to slow down. So that means we're going to need less oxygen because less oxygen is required to pull less electrons through the electron transport chain. So predict the effect of the mutation on the rate of oxygen consumption. Well, the more electrons you're pulling out of these electron carriers, the more oxygen you will need because it's the driving force behind the electron transport chain. Remember, oxygen's super electronegative, so it just pulls at the electrons. So the more electrons you have, the more oxygen you're going to need. But if you're not effectively getting the electrons because your electron carrier is not working, then you're going to have less oxygen consumption. So because your electron transport chain is less efficient, you're going to require less oxygen. And it's less efficient because the electron carrier is not being converted back to NAD plus and H. Justify your prediction, that's what I just talked about. The researcher has hypothesized that the addition of the vitamin that is similar in structure to NADH would increase the activity of the mutated NADH dehydrogenase enzyme in individuals with the disorder. Explain how the vitamin most likely increase the activity. So we're assuming the vitamin is acting like NADH because it has the same structure and it's also producing the same products. So it's basically saying if we increase our substrate concentration, which in this case the vitamin is acting as the substrate, then how could it increase the activity of the enzyme? Well, the more substrate you have to break down, the higher activity of the enzyme you're going to see. So if the enzyme doesn't have anything to break down, you're going to have a low rate of enzyme activity. But if you increase the substrate, which in this case the vitamin is mimicking the substrate and acting like the substrate, you're going to see a higher activity in the enzyme because it has more stuff to do, more things to break down. So that's kind of what you guys should be saying for these. This is kind of a tough one because it puts together enzymes, cellular respiration, and there's always a genetics component that you have to understand what they're even talking about. With the alleles, you have to understand graphs and remember how to do rates. If you have any more questions, let me know. But yeah, hopefully that helps you guys kind of get started on this, and then I can answer any follow-up questions you might have. Bye, guys. Hope you're doing well.